This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Pleasure to welcome Dr. Robin Cash. Robin completed his PhD at the University of Western Australia in 2012, I think it was. And then following his PhD, he spent a good amount of time abroad doing postdoctoral research, both in Canada and the States. And then he returned to Australia to take up a position at MAPRC in, it would have been 2016. Yeah. And I started collaborating with Robin probably about 5 years ago. And soon after that, he was awarded an ARC DECRA, and he took up a position here in our group, and he's been doing some fabulous work in personalising TMS therapies for depression as well as OCD. And recently his technique was approved by the TGA, and it's been clinically rolled out at a clinic in Brisbane. So he's going to present some of his work on translating his technique for TMS optimization. So over to you, Robert.
Thanks. Okay, so yeah, I wanted to, I guess, talk about my research and kind of the state of where things are at in terms of personalised brain stimulation in the field. This is like a really exciting area. It seems like very promising clinically. And yeah, what I'm talking about is transcranial magnetic stimulation. So that's one form of brain stimulation that's non-invasive. So you can see on the right here, a transcranial magnetic stimulation coil. So it's like a figure-of-8 coil. And when you briefly run an electrical current through that coil, it generates a magnetic field. That magnetic field can then cross the skull in a pretty much unimpeded, fairly painless way. And that magnetic field in turn induces an electrical current, and that can be used to modulate neuronal activity. So transcranial magnetic stimulation is usually used, or most commonly used, clinically for treatment-resistant depression, where it's applied to the dorsolateral prefrontal cortex. And response rates are around 35 to 50%. So there's a lot of variation in terms of treatment outcomes. So some people respond very well, others don't respond at all. We get like an average kind of trajectory as shown in this bottom figure of where people are getting better over the course of 4 to 6 weeks. Wouldn't mind going back this quickly. So people tend to get better over the course of 4 to 6 weeks of daily treatment. And then that benefit tends to be sustained. But yeah, this is like an average response. And as I say, some people don't respond at all. So the basis for this variation has been examined a lot. It's obviously very important. And some clinical and demographic factors have been looked at, but they tend to demonstrate inconsistent effects across studies and weak effect sizes. Another aspect that's been looked at is what is the optimal stimulation target site for brain stimulation, and yeah, research in that area has kind of been evolving over the past 2 decades or so. You can see here that different clinical studies and research studies tend to target slightly different regions, subregions of the DLPFC, dorsolateral prefrontal cortex. And you can see here various targeting strategies that have been looked at. So the first two are like scalp-based heuristics, for example, using like an EEG cap and targeting the F3 side that overlays the DLPFC. And let's just go back. And other approaches have targeted specific neuroanatomical regions of the brain, so aiming to ensure that we get the DLPFC. Another approach has looked at targeting regions of hypometabolism in the DLPFC and depression. None of these have really been delivered in any real improvements in clinical outcomes. But recent research using integrating MRI, magnetic resonance imaging, and brain stimulation has started to show that, or it's demonstrated pretty robust and reproducible relationships between treatment response and brain connectivity at the exact DLPFC stimulation target site. Yeah. So in this case, we're talking about functional connectivity. So that's kind of the synchronized activity between two different brain regions. And we're talking about the connectivity between the dorsolateral prefrontal cortex and the subgenual cingulate cortex. So based on this, this obviously presents, this presents an obvious opportunity to improve treatment outcomes by defining the optimal stimulation target according to brain connectivity. One approach would be to take a one-size-fits-all group target that should work best for all individuals. And just to illustrate, so the reason that researchers focus on the subgenual cingulate cortex is that this region has consistently been implicated in treatment outcomes across a whole range of antidepressant therapies. And consequently, it's also been selected as the the target site for deep brain stimulation. So anyway, so there are disadvantages of using a one-size-fits-all group target. So it doesn't really account for the large inter-individual variation in prefrontal network architecture. So you can see that the prefrontal cortex is actually one of the most diverse regions across individuals in terms of network architecture. And this higher diversity corresponds to increased evolutionary development of the prefrontal cortex. So this orange region is showing increased variation across individuals. So these regions vary to such an extent that individual fingerprinting is actually possible based on functional connectivity. And some people have looked at whether a one-size-fits-all group target would improve treatment outcomes, but yeah, outcomes have stayed basically the same. So together, these findings provide a strong rationale for personalisation of the DLPFC target site based on brain connectivity. But an important question is obviously whether precise individualized targeting is even possible or feasible. So when just before we started looking at this, there were basically the kind of like what people were saying in the field was that personalized targeting is not going to be possible. You need like a 20-minute scan. Even then, it's not going to be possible. This paper had just come out, and if personalization was to be possible, you'd be able to identify the same optimal target site defined by brain connectivity, the same target site in across 2 scans in a given individual. So there should be very minimal difference across these 2 sites. So this group looked at that in 100 participants, but the optimal target varied by 3.5 to 4 centimeters across these 2 scans, even like within the same individual. So obviously, this was suggesting that personalized targeting was not, personalized target sites were not reproducible with conventional methodology, not even with the best quality scans. So what we did, we looked at whether we could develop new functional connectivity-based personalization methodology. And we tested this in brain scans from the Human Connectome project to start with. So we had 1,000 scans with participants who had been scanned twice across successive days or even 1 year apart. And we managed to get so we managed to identify reproducible targets using this new computational methodology. So rather than 3.5 centimeters, we were getting down to 2 millimeters, like a difference of 2 millimeters across scans repeated 1 year apart for a given individual. So we also demonstrated high inter-individual spatial variation in the DLPFC. And you can see here that these are 3 example individuals from the Human Connectome project. So you can see that across 2 scans, the target site remains consistent, but there's quite a variation in brain connectivity network architecture. And then something else that was kind of interesting was that we found that we had access to familial data from the Human Connectome project, and we found that personalized sites were heritable, so they were more similar in monozygotic twins compared to dizygotic twins, non-twin siblings, or unrelated individuals. So these sites, this suggests that these sites are stable and under genetic control. So now that we had developed computational methodology to enable personalized targeting, we looked at whether personalized targets were kind of clinically meaningful. And so what we did was we had access to a data set where a cohort of individuals with treatment-resistant depression had received TMS, and we knew they had an MRI, and we knew their stimulation target site, and we computed retrospectively from their MRI the optimal target site. And then we looked at whether individuals who by chance had stimulation closer to their optimal stimulation target had actually had a greater clinical improvement compared to those individuals who had received stimulation further away from their optimal target. And so here you can just see that this is like the variation in optimal targets across this different individuals across the DLPFC. And here you can see that we confirmed this hypothesis, but also this finding was almost immediately replicated by a group at Harvard in a separate cohort as well. So that was reassuring. And now in parallel, quite a few groups around the world have been looking at developing personalized methodology and implementing this clinically. And so some of these studies are really promising. They're all very small cohorts, but the clinical outcomes look good. So just for comparison, so with conventional targeting methodologies, you typically get a response rate of around 46% and a remission rate of around 31%. But in 6 studies to date where they've used personalized targeting based on brain connectivity, the average response rate is 87% and the average remission rate is 63%. So this is huge, obviously. And I should add that some of these individuals had previously failed standard RTMS with conventional targeting, and some of them had even failed ECT. So this is, yeah, it's very impressive, but obviously these samples are very small, and but this line of research obviously warrants further larger clinical trials. This work also parallels similar findings in the realm of the brain stimulation, where some research groups and academic groups have kind of started exploring better ways of targeting the subgenual cingulate cortex. So they've moved from like defining it anatomically to according to certain brain connectivity pathways, and then personalizing this based on individual connectivity pathways, and then refining that further. And as with each of these improvements in targeting, the response rates are now getting quite high in some of the preliminary research. But again, these are small samples, but yeah, it's an exciting time in the field. So far we've had quite a few sites that have been interested in taking up our methodology. So these are some of the sites around the world that are using it clinically or in research. We've been working on developing a toolbox for clinical implementation. So this means that a clinician could basically order certain scans to be performed. These could be uploaded as raw images in, to this pipeline essentially, and the optimal coordinates would be returned. Our methodology has received TGA approval and has been licensed to the Queensland Neurostimulation Centre, which is run by our collaborators and is a non-for-profit clinic where funds are reinvested in research. We're also looking at whether there are differences in adolescent depression or whether this methodology could be extended for adolescent depression for TMS at origin. We have received a TMS machine and we should be receiving a neuronavigation device in the coming weeks as well. So we're developing like a TMS platform to integrate with imaging here. So another interesting question is whether these findings, so whether personalized targeting is actually important just for the prefrontal cortex, or could it be important for other brain regions as well? And the parietal cortex is an obvious brain region to look at. You might have noticed before that this area also has high inter-individual variation in brain connectivity. And there's been a whole series of papers published by Joel Voss and colleagues on enhancing memory performance by stimulating the parietal cortex, which is part of this cortical hippocampal brain memory network. So basically what they've done is people get a brain scan and they do a test of associative memory, and then stimulation is performed, TMS stimulation is performed for 4 days sequentially, and then associative memory is tested again, and then there's also a sham protocol. So we knew the stimulation targets for a cohort. So some people were monitored, looked at replicating this work. We knew the stimulation coordinates for each individual, as well as their change in memory performance. And we basically just adapted our personalization methodology and reapplied it to this kind of scenario, and anticipated that individuals who perchance had received stimulation closer to their optimal connectivity defined stimulation target would have a greater improvement in memory performance. And that's what we saw here as well. And the finding was actually very robust, like without going into detail, it was a very robust finding. So that was kind of cool. Another thing that's worth adding is that TMS tends to modulate like not just one region, but like regions within a distributed network. So this is a previous paper that a master's student led. And basically we targeted the cerebellum and then looked at a cognitive network. And you can see here individual data. This is a change in brain connectivity before. So basically you put someone in their brain scanner, take them out, so do a resting state scan, then take them out, do brain stimulation, put them back in, and look at changes in brain connectivity. And we did this with real and sham stimulation. So with this protocol, you could see a clear decrease in brain connectivity after TMS with the real condition, but not with the sham. And this was fairly, like there was some variation across individuals, but it came out in the group average. You could see the same relation and several different brain regions that were part of the same network. And importantly, in the control region, there was no change. So yeah, it basically just shows that when modulating entire distributed networks, interconnected networks. And so this relates to the next point, which shows that, so this guy at Harvard, Michael Fox, who's really big in the field, he identified that, or he basically did a review and showed that for any given disorder, the most therapeutically effective TMS and deep brain stimulation targets are functionally connected and tend to target the same brain circuit. And one of his students, Sean Siddiqui, he looked at whether this was the case in depression, and looked at it in 14 data sets, including ours, in a total of 713 individuals. And he showed that DBS and TMS therapeutic efficacy was linked to stimulation of the same brain circuit, but also interestingly, lesions that evoked disorder-specific symptoms also impinged on the same brain circuit. So he used this to define this kind of convergent brain circuit that relates to clinical outcomes in depression. And this then relates to my next point. So might seem unconnected, but it will be obvious in a moment why I'm mentioning it. So there was this meta-analysis performed by Simon Eichhoff and colleagues, looking at task-based MRI, so cognitive tasks and emotional tasks in depression over the past 2 decades. And what they found was that each of these studies was giving different coordinates, and they couldn't make sense of it. Basically, there was just no convergence across studies in terms of sites that they were seeing as being dysfunctional and abnormal. So what I did was basically the concept was to take this away from a regional focus where we're looking at spatially inconsistent findings and see whether these sites impinged on or implicated a common brain network. And I did a whole bunch of analysis, including like a whole bunch of null tests, null models, and essentially like this turned out 2 different networks, so one for emotion, one for cognition. But these networks were highly robust, distributed brain networks. And interestingly, if you look at how they relate to previous circuits, like this convergent circuit that I just mentioned, or the lesion circuit that's been previously defined, or also the brain network map of functional connectivity from the SGC, which is used in guiding TMS targeting, there was basically these 2 networks recapitulate these clinically meaningful and independently derived models of depression circuitry. Lastly, I did an analysis similar to what I did previously, which is to identify an optimal brain stimulation target by looking at, by using this network map to essentially extracting the time series of the network map and then using that to define an optimal brain stimulation target. And I basically used the same cohort that we had previously. So this cohort of individuals who had received TMS for depression. We knew their actual site, and I computed the optimal site using this approach. And there's a weak relationship, but it's significant. Actually, it's pretty good, but it's not something that you would necessarily use going forward. But it still indicates that TMS therapeutic outcome related to how well this emotional dysfunction circuit was targeted across individuals on a personalised basis. was kind of cool. And then one other thing that I've been involved in is optimizing the design of TMS protocols. So this is work that I did in Canada. So I guess the point is just to say that so usually TMS is applied in this kind of burst pattern. And early studies, they used 32 seconds between these bursts, which meant that simulators wouldn't overheat, coils wouldn't overheat. And actually that was the main rationale for it. And it hadn't really been revisited since. Anyway, we did research looking at whether this break was actually important at all. With modern devices can now go down to 4 seconds if necessary. And we also used 20 hertz pulse, 20 hertz burst rather than 10 hertz, which allows us to squeeze in twice as many pulses in a given time. So we basically brought down the stimulation time from actually typically above 20 minutes down to 3 minutes. And this is just a response. So we just did this in motor cortex and found that there was no influence at all of the intertrain interval here. And then since then, my colleagues in Canada have examined whether this, or actually demonstrated that this delivers equivalent results in depressed individuals compared to like a 20+ minute protocol. So you can bring it down to 3 minutes and it still gives you the same response and remission rates. It's well tolerated. It's now been implemented in over 10,000 patients across North America. And it actually makes it more affordable for many patients in the States, especially who are denied coverage by their insurers for brain stimulation. And the outcomes are actually really good. So like in this, so there've been studies now in over 400 individuals where more than 70% of patients experience remission from suicidal ideation after receiving this treatment. And there were also high remission rates of 43 to 48% in samples of several hundred individuals. I think up to 900 individuals. Yeah, anyway, that's my overview. So yeah, a lot of this work has been done together with Andrew, all of it actually. And then Luca Cocci, some data from Paul Fitzgerald, and some data from Simon Ackhoff and Veronica Miller. And yeah, it's probably all I really wanted to talk about today.
Thanks for that talk. One of the exciting things here for me is that one of the criticisms of fMRI has been the lack of clinical translation. I think what you're showing here is real evidence of the use of fMRI in a clinical setting. And as we speak now, there are patients in Australia that are benefiting from the technique that you've developed.
Yeah.
So I think that's fantastic in the sense of demonstrating genuine translation.
Yeah.
Are there any questions for Rob?
Yeah.
Chris?
Well, that's fantastic work, Robert. This seems obvious that we should be using sort of these personalised way of targeting when we do TMS. But how, how feasible is it for, like, there's a lot of TMS clinics opening up now, and I guess whether or not they send someone off to have an MRI scan before, just in a normal clinical way of doing things. I mean, I probably wouldn't have an MRI scan. If they were just to go to how can you run this out sort of just in a traditional TMS clinic to make it a practical thing to do outside of a sort of research setting? Like, what are the steps?
Yeah.
Where do you see you need to go to make it just simple to use? And like you say, you return with the Optimism software, you return a coordinate.
Yeah.
Like, how can you make that sort of easy to do and turn around quickly and make it practical?
Yeah, I mean, that's a really good question. So I think for starters, a lot of these clinics tend to be situated either within hospitals or within a broader kind of healthcare network. So it's not too difficult to send them off for a brain scan. I mean, it's obviously used routinely for like Alzheimer's or Parkinson's or bone fractures, etc. So that's not too hard, but yeah, then if they have these brain scans, if they can just upload it to the software, which we're trying to make freely available and readily available, and which will run quality checks, etc. That should spit out the coordinates. What they would then need is a neuronavigation device, which will cost them, but I think they might be able to get it on loan as well, or in installments.
Yeah. I don't think the practicality is who uploads the scan, like the radiologist.
Yeah.
We have to come back to the people to.
I think either the radiologist or the person running the TMS clinic and who would then perform the neuronavigation would have some kind of sense of how this all works. But yeah, I mean, I think it's like there's a bit of reluctance to adopt this because it is a bit cumbersome. But I think like when you see these response and remission rates, I mean, I think it's just, I mean, you'd be crazy not to not to at least give it a go, or at least as the evidence kind of starts to accumulate. Like it, I mean, I feel like patients would, if they're informed, they'll probably be demanding this at some point.
Is it possible because of industry partnership or something that can do the IT side to make it easy and tractable?
Yeah, possibly. Well, yeah, we did think about partnering up with one group. Or alternatively, like running a service where people pay to have these scans analysed remotely if they don't want to do it locally. So that could be performed on a super fast computer and returned within 24 hours or something like that, rather than
the practical stuff. In a sense, is the easy stuff to figure out. It's more about building the case so that you can get, for example, Medicare rebated.
Yeah, that's true.
This sort of scan.
Yeah.
Like the other scan that's not.
Yeah.
Neuro or not.
Yeah.
So what, I mean, it's pick up on the same point, but what needs to happen for the case to go forward?
I think we need, I mean, first of all, I think we need stronger evidence from a large clinical trial. And we're hoping to get funding this year. Like we got very close last year. But if we can do like a large multi-center clinical trial that really demonstrates that this approach does result in much better response and remission rates, then I think that'll be a strong case to present to Medicare.
And then you work through the health economic side of it.
Yeah. Yeah.
Challenging.
Yeah. Yeah. We, I mean, as part of the grant, there is a, we also got some health economists who would be looking at whether this really like adds value compared to like treatment as usual.
Any other? There's lots of applause online, and everyone stayed on to listen to the Q&A, but no questions will come out at this time.
Okay.
I wonder, a question for me would be that here you focus specifically on fMRI functional connectivity. How do you see other modalities such as, for example, the work with CINA on high-risk structural connectomics informing the choice of site or personalising other aspects of the treatment, or for example, the work with Outcon on looking at modelling and the cortical structure. So what are the other factors, imaging or neural factors that could potentially influence the choice of site or influence personalisation in general? So how would you see this going forward in the next 5 years in terms of other factors that would influence the...
Yeah, I mean, I think, it's possible that structural connectivity will add something that's robust and which could help inform the simulation target side. I think functional connectivity has the advantage of just kind of being able to pull out polysynaptic connections. But I mean, structural connectivity can do that now as well, the multi-cloth and so on. So, I think I feel like you kind of alluded to a lot of the next steps around kind of another thing we want to do is kind of take this into native space. So like currently we're working in standard space. We want to take it into native space. So placing like an electrical field or modeling the electrical field on the person's cortex and seeing if that helps us refine the optimal stimulation target and further reduce variation in treatment outcomes. But
and what about criticisms about the TMS, the actual stimulation radius is, it's not focal in the sense of quite. So how, I mean, if we can, we've got precision with imaging to get down to millimeters. Is it a valid criticism to say that do we really need that level of precision, given that TMS is a fairly, the magnetic field is relatively distributed. So what is the?
I feel like that's unfair because you're throwing back at me the exact point I read the other day, but I think it's hard to reconcile like the kind of the importance of precision that we're seeing here with like these massive E fields that we see in some of the models published in certain papers. There's one paper that I found interesting which talked about the spatial extent of TMS-induced activation is remarkably restricted, affecting the spiking activity of single neurons in an area of cortex measuring less than 2 millimeters in diameter. I mean, there's some middle ground somewhere. So something that could be beneficial as well is to use like smaller TMS coils that would be more focal rather than the large ones that are commonly used at the moment.
But yeah, as the technology develops, we probably be able to target in more precise, multi-coil stuff that you mentioned. I was curious if there's any evidence that focus on translating IMIP.
I know there are some groups that are using transcranial focused ultrasound and stimulating the subgenuine cingulate cortex. Not quite sure what their results are, but yeah, that's definitely an area of active research and something that would, I mean, if you could directly target the subgenuine cingulate cortex, you might even be able to just not even worry about all this connectivity stuff, but that would potentially be a more direct way of getting at kind of the core of depression, maybe. Not sure.
Obviously, the first thing that we'll see is that everyone is different. I'm wondering if you have looked whether that somewhat relates to that functional pattern that you're seeing, for example, with the optimal side is always the gyri optimal side. If that's maybe that's even a simpler way to find those regions, like it, I've heard that like TMS is more influential if you put it right over the right over the sulfide. So I'm wondering, is it just whether like the group average location is not necessarily always that optimal location as we put reporting?
I think like our approach won't take that into account just yet, just because we're working in standard space. I don't think it generally will pick up a region that's exactly on a sulcus or a gyrus, but it could be that would be, for example, the boundary of a gyrus or something like that might be more effective, like a more effective site based on E-field modeling. So on. So, I don't know, it's a good question.
Yeah, I was going to ask a similar question. Not just last, but more from again, of course, the benefit from constant counterbalance stuff. But because we know some stimulus cell density region or the neuron structure, they relate to the how the brain will be connected either functionally or structurally. So is that possible, or have anyone in that field so that people say that how this stratification relate to, so how much variance can be explained by this pattern of stratification can already explain an activity pattern. So that is possible, then instead of like pushing the link to more and more imaging modalities or more complicated structure functional activity, if we can just get information from the T1 scan, and we think some maybe more computational modeling, but to reduce the cost of scan itself, then if it already can like achieve some benefit to the certain level. So
I think I would see it as the T1 scan kind of being used to perhaps select between two different potentially optimal sites or kind of refine the optimal target. I don't think the T1 itself could be used to kind of pull out the optimal site.
But how about like maybe the current T1 is not feasible, but like the multi-layer like structure scan where they can get like the neural density information, because when we know that's strongly linked to the activity pattern. Again, I think it would probably help to refine things, but I think like with that kind of multi-layer thing, I think would like that's one thing that we're discussing, maybe including in some of the scans going forward. But hard to say. I don't. And the answer is I don't think anyone's looked at that.
Any more questions for
James King says, great work, Robin. The new study in adolescent depression sounds amazing.
Oh, cool.
I think that probably wraps it up. So once again, join me in thanking Robin for the fantastic. I think we've got lunch. I assume this is for us, right? So we've got lunch here.